So thank you all for being here this morning. My name is Carrie Carpenter. I'm a third-year PhD student here in the School of Plant and Environmental Sciences, and I'm excited to be here today to talk to you about microbial hitchhikers on microplastics. But before I get started, I'd first like to introduce the other team members working on this project. And so from left to right, we have Dr. Schmally, who's not only a professor in the School of Plant and Environmental Sciences, but he's also the director of the Translational Plant Sciences Center here at VT. In the middle is Dr. Fruiten, who's an associate professor of civil and environmental engineering. And all the way on your right is Dr. Gray, who's an assistant professor of biological sciences. So we've come together from different disciplines to try to tackle this issue of microbes colonizing microplastics. So let's get started. Microplastics are commonly referred to as MPs in the scientific literature, and they're unfortunately prevalent throughout our environment. They can be defined as plastics that are less than 5 millimeters in size, and we've been able to isolate microplastics through various ecosystems, including aquatic systems, different terrestrial soils, as well as the atmosphere. And today we're really going to focus on microplastics in the soil. But before we get into our project, let's talk about where microplastics come from. So there are a primary source of microplastics as well as a secondary source. Primary source refers to they were intentionally made that small. So this could be something as the clothing that you're wearing and the different fibers that are thread into the clothing. It could be cosmetic microbeads, which have been banned now for 10 years, but they're still out there in our environment. Or plastic pellets that are manufactured for various different reasons, including for molecular biology and used for drug delivery. So the other way that microplastics come about is via a secondary source, and this is when a bigger plastic or a macroplastic is in the environment and it unintentionally degrades. So there's a few different ways that plastic can break down once it's in the environment. And some of these factors can include physical abrasion. So imagine a plastic floating in the ocean, a wave crashes into it and shoves that plastic into a rock over and over again. There's that physical force breaking down the plastic to become smaller and smaller. The plastic may interact with various chemicals, which then attack the chemical bonds making up the plastic, again, breaking that plastic down further. Different forms of light, especially UV light, can make plastic very brittle. Different temperatures can also create these microplastics, and not just necessarily hot temperatures, but really cold temperatures, and especially thinking about the freeze-thaw cycles and what that does to break down the plastics. And of course, once the plastics are in the environment, they can be colonized by microplastics, including various fungi, which some produce enzymes that attack these plastics and break them down to become even smaller. So, you may have noticed on the previous slide that in the secondary source, I had an image of a tire, and this is because tires are considered a plastic, and tire wear particles are generated when tires are driven on the road, and they create that friction with our road, and tiny little particles can be emitted. And these tire wear particles are considered a unique microplastic. Although tires are not entirely plastic, right? There's also various other chemicals that are included in the manufacturing of tires, including heavy metals, as well as a ozone protectant that when it enters aquatic systems, it becomes toxic to aquatic life. And so we chose to work with these particles as our microplastics in our project, and we obtained these particles from a testing facility in Akron, Ohio, where they do various different testing on the tires before they go into production. And one of these tests is a durability test, where they look at the tread of the tire and how long it takes for the tire to break down. So what you're seeing with the end of the arrow, it kind of looks like black dust, and that's essentially the tire particles that are coming off of that tire as it breaks down. And you can see it visually a little bit better in this flask to your right there. That's about 1 gram of tire wear particles. And so just to illustrate how tiny these particles can get, I'm going to show you some images that I took with a scanning electron microscope. So these are two different images, but you can see that the tire wear particles are not uniform, and they have various different shapes. So there's some fluffier looking particles, and then there's also some bigger jagged edge particles. But what I really want you to notate is the scale bars, which read 500 micron, and that is a 0.5 millimeter, which is approximately as thick as if you took 5 pieces of paper and stacked them on top of each other. So it's very, very tiny. And we want to look at the tire wear particles on this scale because we want to look at what's growing on the surface of these particles. And so once these particles are emitted from the vehicles and they end up in the environment, they can be readily colonized by various different microbes. And I have a few of my lab members are walking around and they're handing out some nerd gummies. As you receive your nerd gummies, I'd like you to use it as a visualization. And imagine with me that the gummy in the middle is a microplastic and the nerds surrounding the outside are various different microbes colonizing that surface. So please use that as a visual, but also enjoy it as a little treat as well. So it's really important to study microbes on the surface of microplastics because once they attach to the surface of microplastics, they induce change to the microplastic surface. So this image above me is another scanning electron microscope of a biofilm on the surface of a plastic, and you can see where the biofilm ends and where the plastic begins. And so once microbes colonize the surfaces, they can ultimately change the microplastic's density, its roughness, hydrophobicity, the functional groups that are available on the surface, as well as the sorption capacity, impacting how it might uptake heavy metals and other contaminants. Okay. So while knowledge in this realm is growing, we still have some knowledge gaps, especially pertaining to tire wear particles and the colonization of tire wear particles. So some of the knowledge gaps that we hope to address with our project are understanding which microbes preferentially colonize tire wear particles, and particularly with our project in the soil. We want to understand how, as seasons change, microbe colonization changes. And then we're also interested to understand how biofilms on the surface of microplastics impact other microbes in the environment. And so to do this, we have a few objectives. So we're going to inoculate soil samples with tire wear particles that are considered pristine, as well as tire wear particles that are previously colonized with a bacterium Pseudomonas syringae, and I will talk a little bit more about that bacterium in a few slides. Once we inoculate the soil, we're going to then compare across seasons by collecting the soil at various months and looking at how the colonization changes, and then of course assess the differences between those pristine tire wear particles and the pre-colonized particles. So I'm going to really talk about our methodology. So this project is in its infancy. We just started our first trial. So I'm really going to focus on what we're doing, and then I'm going to touch a little bit on what we hope to gain and the broader implications for society as a whole. So our soil will be collected monthly from Kentland Farms, and the picture that you see above me is a plot of wheat and barley. These plots are run by Dr. San Antonio, also part of the School of Plant and Environmental Sciences and Plant Breeding. So we'll go out there monthly, and what we hope to gain from this is how colonization changes with the growth of wheat plants. So what you're looking at right now was March, and the plants are about maybe a few inches high, but when we go out there and again this month, they're going to be a little bit bigger. And so the goal is to kind of look at how colonization changes with the growth of winter wheat. So once we collect our soil, which we went about 20 centimeters deep, we brought it back to the lab, and placed it in this cabinet, spread it out really nice, and then we allowed it to air dry by checking the moisture, and once the moisture was in equilibrium with the atmosphere, it was considered air dried. We then sieved it through a 2mm mesh, and this was to homogenize the soil so that we can mitigate any biases of soil that's maybe clumpy, and that might be altering how the tires impact with the soil overall. Sorry. So once our soil was prepared, we then prepared our tires by first passing it through a 5mm sieve, and then whatever came through that, we passed through a 1mm sieve and collected that. So our particles are between 1mm and 5mm in size. Our tire particles were then collected and autoclaved, and this was to kill any live biological material that might already be on the surface of these tire wear particles. So once they were autoclaved, some of the tire wear particles were set aside, but some of the tire wear particles went on to be colonized by Pseudomonas syringae. And you might recognize the word Pseudomonas, particularly Pseudomonas aeruginosa is a human pathogen, and it is a threat to those with a weakened immune system, particularly in hospitals, and they form biofilms on these plastics. But it's much safer to work with not a human pathogen, but a plant pathogen, and that's what Pseudomonas syringae is. So in this, again, this SEM image that I'm showing above me, that's Pseudomonas syringae as a biofilm on a polystyrene microplastic. But to the picture to the right is some of the disease symptoms that Pseudomonas syringae can cause in various different crops. So once we had our different treatments set up, we had to verify that Pseudomonas syringae could colonize the tire wear particles. And if you recall, I showed that SEM image of the fluffy pieces of particles, so this is zoomed in even more on one of those fluffy tire wear particles. And as you can see on the right, there's a nice little biofilm that has formed right in the nook of this fluffy piece of tire wear particles. So now we've verified that it can grow on the tire wear particles. We're ready to start our experiment. So we placed 1 gram of tire wear particles to 1 kilogram of soil, creating a 1% concentration. And so we use these, I think they're referred to as T-balls, to keep the tire wear particles entrained. And so we had 16-centimeter pots. We would put a little bit of soil down on the bottom. We'd have these clasps, and then we would mix the soil and the tire wear particles in the clasp, and then the rest of the soil would surround it. And so our pots and all of our treatments will then incubate in the greenhouse for 1 month. We'll monitor the soil moisture, ensuring that the soils don't get too dry. And then we're hoping to get some data from this. And of course, we will do this, like I said, monthly. So we're going to be March, April, and May. So once the incubation period is over, we will remove those clasps with the tire wear particles. And some of the tire wear particles, as well as some of the soil, will then be sequenced via 16S. And this will allow us to see who is colonizing the surface of these particles, as well as the microbes that are present in the soil as well. The rest of the ambient soil will then be sent off for testing, and total nitrogen, total carbon, and total sulfur content will be quantified, as well as the pH. And so this will give us a little insight into how the tire wear particles impact nutrients within the soil as a whole. So with this, our work has a few different implications for society as a whole. So by understanding what microbes colonize the surface of the tire wear particles, we may get insights into how this impacts soil fertility as well as plant health. Also, by understanding colonization and understanding which microbes preferentially prefer to colonize the tire wear particles, this can lead to understanding soil properties further, which can then relate to a bigger discussion on microplastic pollution in general. And then, of course, if microbes that colonize the tire wear particles shift maybe nutrient cycling or change pathogen dynamics, this can have broader implications on water quality, on biodiversity, and ultimately can impact human health if we're consuming these contaminated foods as well as water sources. And hopefully, our data can help inform policy and regulation, not only in the production of tires, but also tire runoff, and as a whole, pushing for sustainable agriculture to mitigate this new pollutant in our ecosystem. But before I wrap up today, I would also like to talk about how we're not only doing this project, but we really care about teaching the next generation about this bigger issue and hopefully inspiring them to maybe want to get into microplastic research. So some of the activities that we've done are Hokey for a Day. So this picture is actually from a few days ago. We interacted with about 80 fifth graders at the Moss Arts Center, and they got a hands-on experience where we hid microplastics in sand, and they got to dig it out and sift out and find the microplastics. And they got very competitive. They were trying to beat each other and find the most. We also have on the right is a picture of the Pedro Yates Institute at Virginia Tech from last summer. So this institute recruits highly motivated, rising high school seniors, juniors and seniors that come and have an immersive experience here at Virginia Tech. And what we were doing is, in this picture, is I actually grew Pseudomonas syringae on some plastic, and then the students got to dye the plastic to visualize the bacteria, and then they got to view it under the microscope. So we're hoping to continue doing not only these activities, but maybe even more, really pushing home the importance of studying this new pollutant in our environment. So with that, I would like to give a big thanks to our funding from the Center for Advanced Innovation in Agriculture at Virginia Tech, as well as some from the National Science Foundation. And a big thank you to the Nanoscale Characterization and Fabrication Lab, which allowed us to use their scanning electron microscope, and that's how you got to see all these wonderful images. And of course, thank you to my fellow team members and my lab members, and thank you to you for being here this morning and listening. I will gladly take any questions at this time. Thank you.
Open it up for any questions.
Thank you. Have you devised a method to separate the tire wear particles from the soil samples?
So what we're gonna do, which might take a while, is we're gonna take sterile forceps and pick out the tire wear particles. So it is a little time consuming, but as of right now, that's the best way to separate it. We could try sieving it as well, and so we might try that as well. Thank you.
So I really appreciate the definition of microparticles. I see it in the news all the time, and rarely is it defined and exemplified. So that's really great to see. Can you sort of take it to the next level? Is there like a size? You know, it's got to be less than this many millimeters or micrometers or something like that before it becomes particularly troublesome, or is it just sort of anything that's sort of degraded from macro particles? Or well, give me a real definition.
Yeah, I think really a general definition is plastic that is less than 5 millimeters in size. And once it reaches, I believe, 1 micron, then it's considered a nanoplastic. So between 1 micron to 5 millimeters, which is essentially 5,000 microns, that is considered a microplastic.
Well, I try to come up with the optimistic question of, is there any of the microbes that eat the plastics that could get rid of the plastics and be good and wonderful?
Definitely, there's, so I mean, this research isn't focusing on that, but there are many researchers out there today looking at not only setting up experiments to see which microbes colonize and potentially break down the plastic, but with the advance in genomics and transcriptomics, we're able to just look into the genomes of some of these species and see if they produce certain enzymes, which then can attack these plastics, breaking them down further. And some of them can even break them down and use them as a carbon source, so it's potentially feeding good microbes.